Welcome, everyone, to our January 2025 Powerhouse podcast with the United Mitochondrial Disease Foundation. I am Cassie Franklin, UMDF Community Engagement Specialist and a co-host of today's episode. And I am I'm so delighted to be joined by Dr. Philip Yeske, our Science and Alliance Officer. Phil, thank you so much for being here with us today.
It's been a minute.
It's been a minute.
Yeah, I know it's been 6 months or so since I've made an appearance here. So it's really nice to be back. I always enjoy these conversations. And Cassie, I can't thank you enough for the great job you've done continuing to advance discussions with our community, with patients and stakeholders. You've really done a great job with it. So you've set the bar high for me to come back. Yeah,
I know. What have you been up to in absence?
Well, yeah, it's been a busy time, right, for the foundation. And as most people probably know, I've been acting as an interim managing director for the last 6 months here, right, for the foundation, while we've been conducting a national CEO search. That's been a real honor for me to be able to step in and make sure that UMDF is continuing to deliver mission, that the staff feels like they're heard, and that we're still advancing all the important projects we have. I cannot thank my colleagues, Cassie, you included, for all the support during that time. I really think we can be very proud of the way we've navigated this transitional period. But all good things come to an end. And so I think very soon, if not already, we will have a new CEO on board by the time this video comes out. And I can return to having one job title as the Science and Alliance Officer, which, again, our research mission is so important to us, Cassie, right, as you know, and it deserves full-time attention. So looking forward to that here as we kick off 2025.
100%. Like 2025, Phil, it feels like our year, working on communications in January and like looking forward to so many exciting things. I know we have a full slate of spring Energy for Life walks to look forward to. For any of our listeners and our audience out there that's really experiencing January winter and a lot of, I know, snow and ice right now as we're recording. Like,
hang in there.
Yes, hang in there. Spring is coming. We've got at least 6 locations across the country where they're making plans to have beautiful spring walks, indoor virtual walks, walks in their own neighborhoods. So there's a lot to look forward to there.
And lots of ways for people to participate, right? I really appreciate you bringing it up, because whether it's on previous podcasts or anytime I'm speaking right with the community, absent a community that believes in what UMDF is trying to accomplish in our mission, and being willing to help fund that mission, we can't advance it. So we're so thankful for everyone that has contributed, has been a donor to UMDF. If you're seeing this and you haven't had a chance, or it's been a while, we really appreciate continued support. Our mission continues to expand. And I think, as we'll hear through the course of today's discussion, there's so many exciting things going on in rare disease drug development that it's a very hopeful time for the mitochondrial disease patient community. And again, UMDF wants to make sure it's strong and can be there to deliver as much mission as possible to this community.
100%. A big part of our mission. We've already started planning, right? Dr. Yeske, talk about a little bit, sure, if you can. I know we're still in planning process, but Mitochondrial Medicine 2025, yes. St. Louis.
Well, this is internally, as you know, Cassie, we always refer to it as our Super Bowl, right? This is the chance to bring the entire community together, all the key stakeholders, patients, clinicians, industry, researchers. It keeps expanding, right, in terms of even the types of people that are attending the conference. And with that comes what I view as a real responsibility to ensure that we have a very strong scientific and medical program, so that attending the conference, there's a chance to learn. But there's also a very active patient family facing the program that's in development as well for St. Louis this year. So our dates are June 18th to 21st, right? I believe,
correct.
Kathy.
Yes.
So this is definitely the time to mark your calendar, make a hotel reservation if you're thinking about it, because that's always
You don't want to delay.
blocks of rooms, right? Do disappear very quickly. And registration should be opening within the next month or so, right, for the conference itself. So, in addition to a scientific program, family program, we're going to continue with the clinical research pavilion, which has been a big hit over the last 2 years. That's our chance to take advantage of the fact that we're going to have several hundred patients, family members attending, and giving an opportunity to participate in clinical research. So we'll have studies there that you can investigate, decide if you want to be a part of. You can be determined if you're eligible for it. And this is your chance, right, to help advance therapeutic development for mitochondrial disease and just our general understanding of mitochondrial disease through clinical research. So that's going to get bigger and better. I think we have some new twists coming this year, so stay tuned for more details there. And then the other big addition this year is we're going to do a master class on Saturday, which is really a chance to target both clinicians and researchers that are more junior in their careers and ensure that they're exposed to the latest information, bringing people to present that can talk about mitochondrial disease in a clinical setting, but also from a research setting. So our goal is really to ensure that we have the next generation of researchers and clinicians that can continue to advance mitochondrial disease therapeutic development and just understanding in general. So that's going to be a lot of fun to see how that goes too. So that's something brand new for 2025.
That's exciting.
Yeah.
I know, Phil, we've got a ton of just things to look forward to this year when it comes to potential therapeutics, clinical trials. And what I found so exciting just in the last couple of months and looking forward is that we've done 2 clinical trial updates in our Bench to Bedside Talk series. And the fact that we had to have 2 because we couldn't get all the updates in one is pretty impressive. Can you
really good point. There's now so much activity taking place, and we do have to spread it out a little bit, and we're ensuring that sort of mid-year, like December, January timeframe, we get updates as well as at the conference, right, in June in person. All of this speaks to really an emerging area of therapeutic development for mitochondrial disease. And we have a couple of companies that are at the stage of either having regulatory filings or being close to having regulatory filings with the FDA, sort of a very late stage, getting close to decisions around approvals. We have a number of companies that are currently in the midst of running clinical trials at various stages, phase 1, phase 2, phase 3. And importantly, there's a big pipeline behind them of companies preclinical, not yet ready to test in humans, but they're making tremendous strides towards designing trials and thinking about places where you can move into mitochondrial medicine. This is incredibly exciting. It remains a huge challenge, and we should take nothing for granted. But I am personally more optimistic than I've ever been that we're at the precipice, right, of starting this era of therapeutics for mitochondrial disease. And I know today's guest on the podcast will have something to say about this too. And so maybe with that, we should move to that stage of it, Cassie, because we have so much to discuss with today's featured guest. I had to write down sort of parts of the introduction because there's so much to cover here, and it's so impressive. You know this person, Cassie, and we really look forward to having him join us. But This guest today has been formally trained in physiology, experimental psychology, as well as in pediatrics, medical and biochemical genetics. That's a starting point. USA board certified in pediatrics, in clinical genetics, in cytogenetics, and in biochemical genetics. So the professional background, right, is unquestioned. Over 50 years of professional career, half a century, dedicated to advancing these fields. It's so impressive, and I'm really looking forward to the conversation today. Currently, our guest is focused more on the development of novel therapies for rare disease. That's a great connection to what we try to do here at UMDF with respect to mitochondrial disease. Without further ado, I'd like to introduce Dr. David Whiteman. Hi, David.
Hello, everybody.
Hello, Dr. Whiteman.
Yeah, thank you so much for joining us today. And that's quite a resume, right, to get started with. We have a lot to discuss today.
Sure. No, happy to join you. And as you know, and I think some of our older listeners and viewers will know, this is a sort of return for me to both the area of mitochondrial disease and the UMDF. I was heavily involved with both the diseases and the societies back in really 1995 to 2006, when I had a change of career direction that I imagine we'll talk about later on.
For sure.
But for those folks who remember me from when I was working in Maine at the Mayo Clinic and addressing these diseases, hello.
Yeah. Well, thanks again. And Cassie, I know you have some work to do behind the scenes.
I'm going to be behind the scenes. Yes. So I'm going to as our listeners know, I am not the scientist. So this is not my subject area. I'm going to let the two of you have a wonderful conversation. And thank you all of our listeners for joining us today. And I'm going to head on out.
All right. Thanks, Cassie. So, David, as you said, it's a career that spans over 5 decades. Maybe just as a starting point to sort of orient the audience, what led, or the story behind why you became interested in pediatrics and medical genetics as you started to move into your career?
Sure. I mean, this was all a long time ago now, and things have changed dramatically since then. But I first encountered patients with rare diseases as a medical student. Not quite sure why that happened, but it did. I had gone to medical school with the intention of training to be a psychiatrist. I had a background in psychology and clinical psychology, and I discovered I didn't really like psychiatry. But what I came to next was acting as a liaison with the pediatric service in the medical school where I was training. And I quickly became interested in children with developmental disabilities. And following that, I ended up with another rotation in another hospital where I worked with a doctor called Guy Salmon, who had written a book about recognizable syndromes in children with developmental disabilities, sort of the British equivalent of what's known as Smith's recognizable patterns of human malformation in America. And I came to realize that recognizing those patterns, understanding them, was a good way to think about the diseases themselves and how they were caused, but more importantly, a good way to understand how to manage children with those diseases. And I was subsequently lucky enough to have a rotation with a professor of medical genetics who actually wrote the book called Genetics in Medicine. And that kind of consolidated my interest. But he advised me to move to the United States for further training. Britain has a long tradition of scientific genetics, but in those days, there was really no genetic counseling. There was no medical genetics physicians. And so I repeated what he had done 15 years later and came to America into one of the first training programs in medical genetics. And over the years, my interests shifted, I think is probably the right word, from what we call dysmorphology or syndrome recognition into newborn screening, more precise genetic diagnosis, in those days using biochemical tools,
Right.
and some early DNA work. And ultimately, in the early 90s, I became interested in more areas of metabolism as a concept, and recognized that in many cases, they actually helped us understand the basis of disease. And so the rest of my career from that point onward became focused on biochemical genetics. And I quickly developed two major interests. One was mitochondrial diseases, and the other was what we call lysosomal storage diseases.
Right.
So
Right.
that's kind of how I got here.
In short, yes. But we'll talk more about mitochondrial diseases as we go forward here. It's such a fascinating time to be active in the field, right? As you described, sort of this move from syndrome diagnosis just based on patterns of symptoms, right, to having some biochemical tools that could advance, right, a diagnosis of the patients. And then, as we turn towards the 21st century, increasing use of genetic sequencing, right, to be able to get to a diagnosis, right, for these patients. It's been a pretty incredible journey, right, over those major paradigms during your professional career. So maybe looking back over now some 30 years that you spent in academic or academia, and then 20 some years in more of a medical industry, pharmaceutical, biotech. What are some of the key points you look back to, sort of the inflection points, if you will, that kind of remain with you over the course of your career?
I think the important ones are the better, first biochemical and then subsequently genetic understanding of disease. Yeah. This applies particularly to mitochondrial disease.
Yeah.
At the time when I was working with mitochondrial disease patients back in the 90s and early 2000s, apart from a few, shall I say, obvious examples, the diseases were not well defined biochemically or genetically. There were certain biochemical characteristics that we all used to characterize, to, if you will, confirm is too strong a word, but to focus on the diseases. But those were mainly clinical presentations, and essentially this was a lack of energy. And we saw two kinds of that. One was where we saw it, and people have since proven that there is mitochondrial dysfunction in a variety of other genetic diseases and syndromes, and that's an important thing going forward. And it's also triggered a lot of the move towards therapeutic development. We can come back to that later. And then the others were children, mainly, some adults, who we either felt strongly or in some way had been able to confirm had what we used to call primary mitochondrial dysfunction, primary mitochondrial disease. I think that was an important starting point. Subsequently, other important milestones have been the precise genetic delineation of those diseases. I mean, there are so many different mitochondrial diseases right now that somebody wrote a book about them, and I can't remember them all in my head. Some of them I recognize as things I knew differently before. Others of them are total novelties. And then there is the approach of using that information towards either what one might call empirical biochemical therapies, specific vitamins, cofactors that might help an individual disease, or in the longer term towards gene therapy, either by gene replacement or gene editing. In the old days, we simply used to refer to, quote, the mitochondrial cocktail, which was a collection of vitamins and cofactors that we administered very empirically to patients who we thought had these diseases. Sure. Some of those patients responded quite dramatically, and we now know that's because they had a specific mitochondrial diagnosis that, for instance, required supplemental riboflavin. Others of them actually didn't respond, but it was all we could do, so we did it.
Well, they still remain standard of care, right, for mitochondrial disease patients, absent approved therapeutics. And it remains a really important component, right, of how our patients are cared for. We hope that will change over time, right? We'll have more targeted therapeutics.
It will, yeah.
Yeah, for sure. Well, I know, as you moved into 2000, in 2017, you were appointed as president of the board of the American Council of Medical Genetics, ACMG. And you at that time really emphasized education, right, as sort of the platform that you wanted to bring to it. And, as I kind of went through your bio and looked at all the things you've been involved with, clearly education, collaboration, mentorship has really been important to you. Maybe just speak to sort of your philosophy on why that's so important.
Well, I think as one of the first people to work in these areas, as things developed over time, partly just as a person and a pediatrician, but more importantly, as a geneticist, I felt it was important to continue to train and educate the next generation. I'd actually ended up in industry by accident. Some people know this probably, but for family reasons, I had to relocate from Minnesota to Boston in 2005. And I actually couldn't find another academic job. I was at that point in the academic hierarchy where there were not very many positions for someone like me.
Right.
And after some discussions with friends and others, I ended up becoming a medical director for actually a program for an MPS disease called Hunter syndrome in a small biotechnology company. I thought I'd do that for a year and then go back to a real job once I'd found one. But that didn't happen for 5 years. And by that stage, I was really keen on drug development. And in a sense, it was the next stage in my life, even though I didn't consciously know it as I went into it. And through that, I was able to organize a number of medical programs around, well, obviously that particular disease. It was always my dream to have that company start doing research in mitochondrial disease. Unfortunately, for essentially commercial and economic reasons, that never happened. The one time that company actually looked at a treatment for mitochondrial disease, which I shouldn't mention by name for legal reasons, they actually decided to pass on it. Unfortunately, another company that you probably know well actually made that into a real treatment that's helped a great deal of people and helped that company. But that was unfortunate. That's how decisions get made in business.
I was just going to say, it is kind of the nature of therapeutic development that decisions are made both for scientific reasons as well as business reasons. And it can be very frustrating, I think, particularly for the patient community, right? That, that's the common thread through all of it. Nothing has changed. The patients still need those therapeutics. But the challenges, right, associated with being able to develop therapeutics force some difficult decisions sometimes that aren't necessarily the result that we hope for.
We want hope for, yeah. And we can return to that theme later on when we talk a bit about the future. But a little after I joined that company, I was fortunate to move into a position where I had a lot of control over the company's activities in regard to medical education and patient outreach, which suited me fine. And then I was able to, I was asked to join the American College of Foundation leadership team and made that a theme there. I was successful at one point in raising quite a lot of money for education through that organization. And this wasn't specifically mitochondrial disease, it was really medical genetics and genetic counseling. But that's been, it's been an important part of my career all the way through.
Yeah, well, certainly at that phase, there's no question. But I think if we look back even further, we know there are many clinicians, researchers who you were a major influence on, through the early stage of the career. We reached out to a number of them just to get thoughts and comments. And one of them said, I'd really like to be able to join. So we'd like to bring a special guest into the room, Dr. Whiteman, someone that you've had a chance, you've definitely had an impact on. And all right, Cassie, why don't you bring our mystery guest forward?
Oh my gosh. Hello, Jerry.
I feel like a game show.
Yeah, I know. We should have done like 20 questions or something there, right?
This is your life or something.
This is your life. There we go. Well, yeah. So as we saw on the screen, Dr. Jerry Vockley, certainly well known in the mitochondrial disease community, a current member of the UMDF Scientific and Medical Advisory Board. Jerry, thank you for your many years of service there in that way. But we know you have a deep connection to David, and he was a big influence early in your career, and thought it would be nice to maybe share some of that with our audience.
Yeah, boy, I think I've known you for 45 of those 50 years, David.
I think so.
We go back. David was a fellow in Dr. Bill Melman's lab at the University of Pennsylvania in the Department of Human Genetics. He was a genetics fellow at Children's Hospital in Philadelphia. When I joined the lab of Harry Harris as a graduate student, also a faculty member in human genetics. So David was probably the first person I ever recognized as being a medical geneticist, and I thought, well, that's kind of interesting. And how do you do that?
It sparked a thought. Right?
What is that? And it really switched on the light bulb, and I feel like we've been doing this the rest of our professional lives. We've crossed paths a number of times, including being on the faculty together at the Mayo Clinic in the in the early 2000s?
Early 2000s, Jerry.
Yeah. A great opportunity for our kids to get to know each other and strike up a friendship with Mary Ellen, David's lovely wife. So it's been great opportunities professionally and personally for me over the years. So I was really glad when I joined the board of the American College of Medical Genetics to I'm working from home. We get a guest appearance from my dog.
Another special guest, yes.
That's right. The issue here is he's going to start poking me to let him outside, but he can wait. But we've had the opportunity to work together at the college and the American board, and lovely to be able to connect again through UMDF.
Yeah, David, I know you saw patients, took care of patients, for many, many years, earlier in your career. And Jerry, you're still very much an active clinician and, someone that sees a lot of patients with rare diseases, including mitochondrial disease. So much has changed, right, over the course of those 30, 40 years, but it strikes me as something much more interdisciplinary now than it used to be. And I wondered if maybe each of you could just kind of briefly touch on that aspect of how you've seen clinical care change for rare disease patients over the years. David, did you want to start?
Sure. I think that the biggest change for rare disease patients in general is, and it's a shift also within professions, is a recognition of what some people call the patient as partner or the patient as part of the team. I was originally trained in medicine in Britain, where there were patients and there were nurses and there were doctors, and that was it. And you didn't necessarily talk to each other, and certainly not about development of therapies. Over the years of working in the US, first in a pediatric residency and then in genetics training, I came to appreciate that it was necessary for everybody to be part of the team. And this applies to all the diseases that I've worked on. Some people bring different things to those teams, and in working in industry, one runs into the same phenomenon. You meet people when you join industry who have professional backgrounds that you've never experienced before, and you don't necessarily think you need to deal with. Most prominently, obviously, the commercial teams. On the other hand, commercial teams exist for a reason, and without them, you'll never finish the product, and you'll certainly never get it to market. Another example would be people who are trained in what many people call the science of drug regulation, mainly people ex-FDA. So I think that the biggest shift is from me as an individual to me as part of a team, and more recently as leader of teams in drug development.
Sure. Right.
And when I first joined industry, my first boss, who unfortunately had a stroke 3 months after I joined them and was not able to work again, but I remember him saying to me on my first day, he said, "David, you've been taking care of the patients. Your job here is to take care of the drug for all of the patients." And that's kind of been the way I've followed things since.
Yeah, and I think your career definitely went that direction. So, Jerry, maybe your thoughts on how you've seen the care of rare disease patients change over time.
Yeah, so rare disease patients have a tendency to fall into two categories. One is everybody else is afraid of them, and so geneticists end up being the ones that take care of them. The other ones are the ones that are so complicated that they're seeing so many different specialists that what you end up having is that old story about the blindfolded individuals trying to feel an elephant and describe what the animal is. So everybody can tell you about their part, but there's nobody there doing the integration. And so for those patients, that's what the medical geneticist is, the biochemical geneticist is. We're there trying to integrate and to use the good old sports metaphor. We're in Pittsburgh, of course, is that you're the quarterback for the team, not so much because you're the leader, but you're the one who's responsible for seeing the big picture and try to make sure that everybody is in fact playing as part of a team rather than just doing their own individual thing. Because sometimes what you do on one side of the patient equation impacts the other side in unexpected ways, and if you're not watching both sides, it's a problem.
Yeah, absolutely true. And you know, Jerry, I can remember a conversation that you and I had a number of years ago about just, with 7,000 plus rare diseases, genetic disorders now identified, as a geneticist, you felt really confident that I can recognize these patients, I can diagnose these patients, but it's so complex and so many disorders now that, you really have to turn to tools like sequencing, right, and genetic diagnosis, earlier, as opposed to maybe more of a confirmatory step, right, in clinical care. And that's been a huge change, I'm sure, that you've seen over the last 10, 15, 20 years.
Yeah, David, there were maybe 200-300 in the textbook when we started.
Yeah,
big difference.
We really didn't know what was beyond the horizon, right? And it's only through science that we've now been able to really discover and characterize all these other disorders. Well, gentlemen, we have a lot more to talk about rare disease, mitochondrial disease, therapeutic development. We want to get to all of that. We're going to take a quick break so that UMDF can provide a message, and we will be back after this short break.
The United Mitochondrial Disease Foundation's Mitochondrial Medicine Conference brings together researchers, clinicians, and patient families to work toward better outcomes for patients with mitochondrial diseases. Save the date and make plans to join us at Mitochondrial Medicine 2025 in St. Louis, Missouri.
All right, welcome back to our conversation with Dr. David Whiteman and our special guest, Dr. Jerry Vockley, who's popped in to join us, and really the two of you to be here and really help advance a discussion around rare disease and just everything that's been learned in this field and how it's changed over the years. David, I know you mentioned, early on, newborn screening became sort of a focal point for you. Maybe briefly tell our audience about how newborn screening kind of changed the game, right, with regards to diagnosis of rare disease.
And it's still changing. I became involved with this essentially of necessity because both in Massachusetts and Maine, I was the clinical geneticist who was dealing with all of the patients who were coming out of newborn screening. So I ended up being drawn into what were then called governor's advisory committees for newborn screening that regulated the screening in those two states. Likewise, when I moved to Minnesota, although other people, including Jerry, were involved there as well. And, newborn screening was not actually new at that time. It had started way back in the 1960s for certain select diseases, not all of which were necessarily even biochemical. But there was an explosion in the early 1990s, really late 1980s, when mass spectroscopy as a diagnostic technique in the laboratory became available in the United States and quickly adopted. And that led to a much greater number of conditions that could be detected, although individual states made decisions about whether or not they wanted to see that data, based largely on whether they had the capacity to treat the condition. And then also many conditions which we'd not really known about before essentially came out of the woodwork, to use an old English phrase, and we learnt about them as we went along. Some of those were milder forms of diseases we already knew about, and some of them were completely new diseases, particularly in the area of fatty acid oxidation, which Jerry can speak to much better than I can. So that led to changes, particularly in the public health community, about how states were assessing and then proceeding to adopt these screenings. A federal committee was formed, a recommended uniform screening panel was formed, which not all states follow as completely as others, and a process is in place. More recently, with the advent of first exome screening and now whole genome screening, a whole discussion has evolved about a paradigm shift there into screening for children directly by analysis of genes at birth. Some people object to that for political or religious or other reasons, but in a practical sense for these kinds of diseases, and I would argue particularly for mitochondrial diseases, this is going to be the way that we go. So let me hand over to Jerry and see if he wants to correct any of that or add to it.
Jerry, I'd love to hear your thoughts on, particularly, moving forward. Is genetics the way of newborn screening in the future?
I would say that we are going to be dragged forward. Many in the field kicking and screaming towards the use of genome sequencing for more broad-based identification. It's really the only way. Many of the diseases don't have a biochemical phenotype or something that you can recognize by just a look at a drop of blood by a simple or otherwise methods. And so while change is difficult, this is a change that's going to happen. My crystal ball runs out after about 6 months. So I don't know if it's going to be 3 years, 5 years, or 10 years. But it's coming, and it's going to be to the benefit of kids. And this is where, as David points out, we're going to identify mitochondrial disease early, and as new therapies come on board, mostly the sooner you treat, the better, and so it's going to be a huge benefit for the UMDF community.
Yeah, so this is probably a great point to kind of pivot and focus right on mitochondrial disease. And, Jerry, maybe I'll just stay with you for a moment. You've already, spoken to the fact of why this is so important to the mitochondrial disease patient community. Maybe just speak to some of the challenges associated with, identifying, characterizing, diagnosing mitochondrial disease. And you have 30 seconds. Go.
30 seconds. Well, I think the easiest way to explain it is that mitochondrial disease can appear as almost anything in anybody at almost any time of life. Other than that, it's easy. And so, it's you have to think about it in most cases to diagnosis, and all too often it's not thought about until very late in the course of the disease, and that's not going to work if you want to have effective therapies. You have to get in there before symptoms start in many cases, and I think it's true for mitochondrial disease to have maximal effect, and so that's why a process that allows you to identify individuals before they become symptomatic, or at the very least, as soon as they become symptomatic, it brings the diagnosis forward to you and allows you to start a therapy sooner rather than later. It also provides the impetus, and I think David can speak to this, for pharmaceutical companies now to say, oh, well, now we're identifying these disorders in a timely fashion. Maybe we should think a little bit more about developing or helping to develop drugs, work with our academic partners to develop drugs to treat them.
Yeah, David, I'm sure you can. Speak to the importance of sort of defining a targeted population, right, that can benefit from any given therapy.
Oh, absolutely. And this is important both from a scientific and from a commercial development perspective, and I'll touch on both. But quickly before I do that, I think there's an important aspect of newborn screening in terms of properly defining the spectrum of diseases and helping in therapeutic development. For a very long time, the golden rule of newborn screening is that you don't screen for things that you can't treat. And there are good reasons for that. I'm not disputing them. But when we have so many individual rare diseases now defined at the genetic level, it's actually important to find those children. It's also important, I believe, to the families. I've always felt that families have felt much more that they want to know whether there's something available to treat or not. So there's an important shift that's been happening in the pharmaceutical industry in the last 5 years. Some people think this is cyclical, and I hope it is. But I was fortunate enough to get involved with drug development for rare diseases at a point where very few companies and people were doing it. And they developed a different model that allowed development of drugs in an economically feasible way for small and rare disease populations. Classic diseases such as Fabry disease, Gaucher disease, Hunter syndrome, Hurler syndrome, etc. None of those are mitochondrial diseases because we didn't even have the capacity at that point to understand mitochondrial disease well enough. But that model worked. But over time, as what commercial people call the low-hanging fruit, which probably sounds like a horrible term here to everybody else, but I'm going to point it out anyway, was achieved. Doing drug development for rare diseases became more complicated, more expensive scientifically. And accompanying that was a shift in the thinking of people who lead pharmaceutical companies away from primary research, primary academic research and development, and more towards what they call late-stage development. What that means in a practical term is that if you're a big pharmaceutical executive, you don't want to buy the initial research out of, say, Jerry's lab or somebody else's lab and move it forward in clinical development. You want to buy something where that work's all been done, all the early, quote, de-risking has been done, and you're looking for something that is, quote, phase 3 ready and can go straight to market in 2 years. And that has really hurt the rare disease community. There are a few companies, I guess I shouldn't use trade names, but most people will know who they are, who are still directly focusing on rare diseases. But the majority of what most people call big pharma, the large companies like Pfizer and Merck and so forth, have moved away from that area a long time ago. The mid-sized companies like Sanofi and Takeda, where I used to work, have done the same thing recently. And essentially, we have to shift a lot of early development back into the academic community, the patient community. I've been pleased to watch how the ability of the UMDF to sponsor research has grown over the years. When I was first involved here, I was on the SAB in the early stages. We didn't have much money. We were doing what most people call seed research or pilot projects. But now I see you sponsoring major development projects. And I don't know the answer to this, because in the end, it comes down to where does the money come from. But I'm glad that the UMDF is stronger in this regard. But there's really going to be an issue going forward about finding money for rare disease research. Now, one of the things you can do to counter that is to use newborn screening and other clinical techniques to find the patients, catalog what they have, catalog what's called the natural history of the disease, and have people and understand in a numbers sense what the patient numbers are, and therefore what the economic potential is. And in other words, play the big companies at their own game and generate that data and then take it into them. And I'd be interested to know what Jerry thinks about this approach as well.
Yeah, I like a hybrid approach. And I'll explain what that means. You're right, David. The companies sort of don't know what they don't know.
Yes.
And so when we talk about rare disease, remember the FDA definition of rare disease is 200,000 people in the country. And we're talking about diseases where there may be as few as a handful. And so you have to help the companies find that sweet spot where there are enough patients where, this is a capitalist society. They have to make money or they're not going to invest in it. So you have to convince them that there's enough need there. And sometimes that means bundling diseases together, So here's where it's useful that as opposed to a problem, that mitochondrial disease is actually 400 diseases instead of just one. When you bundle them together, it ends up being of high enough incidence that it makes it an attractive, if difficult, a target for pharma. Where I think pharma has made a primary shift is from doing their own basic science research to relying on the academic community to do the basic science, and then trying to go in and identify the best targets based on that. And what that allows is I call it intelligent design instead of random design. And those of you who are of an age that you have as much gray hair as the three of us do know where that comes from. But
rational
design, yeah, it might be less. But that is, if you want to develop a hypertension drug, you take 10,000 people and you treat them with a bunch of different drugs and you see which one works best. In rare diseases, you can't do that. So what you have to do is get a much better understanding of the basic defect at the cellular level, and then develop drugs that counter that problem at the cellular level. So in the case of mitochondrial disease, improve energy development in a cell, energy production. And if that works, then you can go to a mouse model or some animal model if it's available, but it shortens the timeline from the idea to something that's ready to go into an individual. From it can be from decades to it's still years, but it's still a much faster timeline. So long as those individuals who are working in the laboratory have the funding that they need to do those experiments, with the NIH budget being stretched thin and diffused out towards what they view as more common, more needy problems, it becomes the role of organizations like UMDF to try to help augment, jumpstart some of those investigations that can lead to the next generation.
I do think this is a place where appropriately resourced patient advocacy groups can play at that interface, right? Between the academic research and sort of the needs of industry, right, to move it into clinical development. That's certainly been a focus here at UMDF with respect to mitochondrial disease. I think we're going to start to see the fruits of that labor here in the coming years, and that's really exciting, I know, for our patients. Gentlemen, we're going to take one more break before we come back for our last section, and we're going to talk a little bit about some of the challenges still that exist for rare disease drug development, but also try to end on sort of future directions and what you're both most hopeful about. So we'll be back after this quick break.
Thank you for listening to the Powerhouse podcast. Join us as we take every step toward treatments for mitochondrial diseases. Together, let's energize the fight. Register online at energyforlifewalk.org. Now, back to the Powerhouse.
Welcome back to the UMDF podcast, where we're celebrating the professional career of Dr. David Whiteman. We're also joined by Dr. Jerry Vockley from the University of Pittsburgh, who trained and worked with Dr. Whiteman early in his career. Both these gentlemen know a lot about rare disease, drug development, diagnosis, drug development. David, we've seen a lot of changes over 40, 50 years, right, professionally. And I know one of the sort of watershed moments for rare disease drug development was the passage of the Orphan Drug Act back in the 1980s. And maybe just you share with the audience how that sparked innovation, but maybe 40 years on, where we need to think about some changes, right, about how we get drugs approved by the FDA, particularly for rare diseases.
Absolutely. And I think it's important to mention that the Orphan Drug Act was driven by a leader of the rare disease community, actually one of the founders and first leaders of the National Organization for Rare Diseases, and I think he was a Senator Udall, who realized the importance of what Abby, and I've forgotten Abby's last name now, was driving towards. And small biotechnology companies like the one that I joined when I first came into industry couldn't have existed without the provisions of the Rare Disease Act. Firstly, they streamlined some of the FDA requirements for approval of drugs, and they offered certain incentives, financial incentives, that were attractive to the people who are running these companies. And that's still important. However, the economics, as I mentioned earlier, of drug development have changed to the point where that's not the key issue now for rare diseases. The changes that were introduced following that into FDA regulatory processes were really at the discretion of the then commissioners of the FDA. There's a very strict rule written into the Code of Federal Regulations and ultimately deriving in a law that requires 2 well-controlled clinical studies to show success to approve a drug for marketing. One of the early changes was that was reduced at the discretion of those commissioners to 1 study. And that's been the gold standard for most of my professional career. Only for rare diseases, not for other diseases. More recently, there's been a move, particularly in rare disease, and this links into what we were saying about newborn screening and genetic diagnosis, towards biomarker-based clinical development. And Jerry and others have been involved in the public discussion about this, because in these individual rare diseases, and we're now getting to that level in terms of biochemical understanding of individual mitochondrial diseases, the chemistry is so clear that the proposition that if you increase the concentration of X or decrease the concentration of Y related to the biochemical pathways of the mutation, you are very likely to achieve clinical success in the disease. However, for a long time, the FDA has resisted that. I'll just digress a minute to Hunter syndrome, where when I first started working on Hunter syndrome, there's a very clear biomarker for Hunter syndrome and some very well understood biochemistry. And we actually used that to go through the drug development process. That was our metric for our success. However, when it came to applying for what's called a marketing authorization from the FDA, we ran into a brick wall because they simply were not willing to consider that. They wanted their well-controlled single clinical trial and a group of ancillary evidence, sometimes referred to as the totality of the evidence, before they would approve the drug. But looking back on it, I could have predicted, I did predict in my own mind that the use of that biomarker would in fact predict clinical success. However, because of the small number of the patients with that disease, it literally took 10 to 15 years post-marketing to gather the clinical data that in fact prove that proposition. And anybody who knows that disease and that drug will know that now. So what's happening at the moment is that people who work with these diseases, both in academia and in certain drug development companies, are pursuing that proposition, partly based on that example, actually, and saying, no, we should approve based on demonstrating a significant change in the biomarker, reducing the concentration, increasing it, whatever, and then commit to what are called post-marketing studies by the FDA, or phase 4 studies that will eventually prove the proposition. Because with the small numbers of rare disease patients, and for some mitochondrial mutations now, there are literally just 5 or 10 patients.
Right.
You're never going to be able to prove that mathematically in a clinical study. So that's where
we stand at a point like this with the Barth syndrome, community right now.
Absolutely.
Mitochondrial disease.
Yeah, and I followed that story carefully. And I think everybody in this community believes that should be approved on that basis, whether it will. And there's another disease going in front of the FDA shortly to treat another MPS disorder called Sanfilippo syndrome, where the same is being attempted. We have to see whether the FDA will actually do that. Sure. Now, in some public statements and discussions, some leaders of the FDA have, I think, hinted is probably the best word, that they would accept such applications. But that's where we are now.
All right. And Jerry, with 400 plus mitochondrial diseases, very rarely do we have a clear biomarker like Dr. Whiteman just described. There are some exceptions, but for the most part, we don't. And so that's the challenge then of designing trials, right, that can gather data to prove efficacy, obviously be safe as well. What do you think are a handful of the most important things that you would like to see the FDA do better to be more accommodating of rare disease communities like mitochondrial disease to be successful in getting to approved therapeutics, absent clear biomarkers?
Yeah, I think that the number one thing that the FDA can do is recognize the validity of study designs other than the classic double-blind placebo control study. That's the design that's classically been used. You give an individual the active compound or a sugar pill. You follow them over time and you see if they're different. And if they're different in a positive way, you call that a successful trial and you approve it. But there is nothing in regulation or law that says that a study has to be double-blind placebo.
Correct.
And in fact, there are lots of very unique study designs out there that have been utilized and or proposed that can get you to a scientifically valid endpoint, a clinically proven outcome that don't involve a placebo control. But they're very difficult. It's very difficult to get the FDA to buy into those, even though when you talk to them in upfront, they they seem enthusiastic about them. When they present on this at meetings, they're very enthusiastic about it. But then you get a bunch of cardiologists who are used to 20,000 individual clinical trials, and they're asking you, what's your control? What's your placebo? And gee, that N of 100, which represents the entire rare disease population in the world, seems small. Which is, where Barth syndrome ran into some criticism. And so there has to be some flexibility there, and the recognition that you can use these alternative designs. When you don't have a biomarker, first of all, I agree with David. Biomarkers are absolutely clear. We have so many examples in our diseases where there's a biomarker that unequivocally predicts outcome. And it appears to be unequivocal except to everybody at the FDA. And they argue about it. And so, what we need is some sort of conditional approval where you can approve it on a biomarker or some sort of smaller study. And then they all do post-marketing studies anyway. If it's really working, then after a few years you take the provisional off of it and you're good. If instead you've made the wrong call and you say, oh well, gee, those initial studies didn't actually represent what longer term or larger population would show, well then you pull it off the market and everybody goes back to the beginning. For something like mitochondrial disease, oftentimes you'll hear talk about a sort of a multi symptom or a multi outcome kind of study, where instead of saying everybody's going to be judged by the same outcome, you're going to take the individual and whatever it is that's worst in them, you're going to look to see if it gets better. And that may be different across the disease. And this is especially important for something like mitochondrial disease, where everybody's a little different.
Yep.
And so you have a suite of things. You look for one or two indicators that are the worst for that individual, and then you follow those over time instead of saying everybody has to meet every parameter in a multi-outcome kind of design. We had a situation where a drug recently went up and for approval, and it was better in, it was like 10 of 12 tasks that they assigned, and the FDA didn't approve it because it didn't meet the endpoint with those other two things, and it was just. It just didn't make any sense if you stopped and looked at it from a clinical standpoint. So having people involved who understand the diseases at the level of the FDA is critical, and they have a new panel that's supposed to do that. But the problem is, and in fact, I've been appointed to it. But the difficulty there is that it's such a small clinical community that most of us are involved in all of the clinical trials, and so we all have conflicts of interest. Yeah. So even though we're supposed to be on these panels, when push comes to shove, we get shoved off because it's not a there's a conflict.
Well, I think we'd all agree. No simple answers here. But it's good to see that there's a handful of specific cases where the FDA has demonstrated regulatory flexibility, and that increasingly the leadership, right, of the agency speaks in a way that there could be even more flexibility, right, going forward. And the proof is in the pudding, and we all know that the mitochondrial disease patients can't afford to wait, and that the real human cost of having to do additional trials, all right, is significant. And we at the end of the day, that is the reason why we're all doing this, is to try to get helpful therapeutics, into the hands of the patients as quickly as possible. This is a topic that definitely warrants much more time and a deeper conversation. And unfortunately, we don't have a lot more today. Dr. Whiteman, I'd like to kind of end the podcast with just hearing some of your thoughts on a couple of topics. One, if you had the Wayback Machine and you're a young clinician, doing a fellowship and starting out, what would be the one thing you would say to, sort of up and coming clinicians now about being involved in rare disease patient care?
Well, for that, I think my advice to them would be to carefully choose either a group of diseases, one disease that they're going to dedicate themselves to. Jerry and I grew up in an era where there simply weren't enough of us who could deal with one diseases. I've been referred to in public meetings before as a jack of all trades, because over the course of my career, I did a lot of things in clinical genetics. These days, there are specialists for all of them. I wouldn't dream of doing some of them again. But for a young person entering the field now, I'd say, with discussion with your mentors and teachers, take a particular disease or a group of diseases, perhaps in the mitochondrial context, and focus on them. But focus on them in getting involved with the patient community, not just by applying for grants, as they might need to do to get their project started, but actually meeting with the community, listening to the community, and then having a solid focus on that moving forward. There's a bigger issue there, which really follows on to our previous discussion. So if you don't mind, I'll just touch on that. Through the 20 years that I've been involved with drug development, what has struck me is that people have always looked at single trials, so-called pivotal trials in industry language. But really, you have to think about this as the overall development for treatment of the disease. So you start with the disease. And I think we need much more integration of what people call natural history studies or natural history registries with particular trials that will always have to be done. And with, more importantly, what most people call post-marketing follow-up, but I'd rather call long-term follow-up. The things that I've learned from the diseases that I've worked from have carried all the way through that spectrum. And development would have been better if we had had more involvement of the patient communities early on in those projects. The traditional pharma model is to do your so-called preclinical development and early clinical work without really consulting the community. That's turned out to be a huge mistake for a number of diseases. A recent example is actually the last disease that I led in my industry career. This is a well-known disease with a terrible outcome. And we ran a development program that worked beautifully from a biochemical perspective. However, for a variety of reasons, some almost political and commercial, the clinical trial didn't measure certain outcomes. And part of that actually was contributed by the FDA, so it's a very complex situation. But the trial focused narrowly on what most people would call survival of those patients, not just survival in the length of life sense, but survival in terms of their ability to move and walk. From that basis, the trial did not succeed. So it was a biochemical success. It was a genetic success, if you want to call it that. It was a technical success from every angle. But when we analyzed the data, the so-called primary endpoint was not met. However, the families and all but one of the physicians who've been involved as principal investigators quickly pointed out that there were certain things that they had observed in the children that were never measured by the company. And fortunately, the company has done the right thing and continued to supply that drug to those patients because there's nothing else available for them for the time being, and there probably won't be for another 5 years. But that would have all been avoided if a better understanding of the natural history of the disease, which from an academic perspective actually existed, although it was in older literature going back 15 years, had been applied to the whole drug development process. And I can see that happening with many of the rarer mitochondrial diseases. You've really got to talk to the families, understand what their goals are, understand what their needs are, and as part of the process very early on. I think and I just want to put a plea here. I know you have such a registry, but to everybody who's listening, I would say get involved with your registry and contribute your data, not just for the sake of your own self or your own child, but for the sake of the mitochondrial disease community.
No, well said. Say that all the time. It's a very selfless act, actually. Even participating in a clinical trial, it may not be to your benefit, but you are making a huge contribution, right, to the rare disease community when you're a part of clinical research and a part of patient registries. Our goal is to try to develop sort of clinical research ready cohorts, right, through patient registries. And that's going to be a big part of the success of getting the numerous therapies that it's going to require, right, to address something as complex as mitochondrial disease. Gentlemen, I can't thank you enough for taking the time sharing with us today your thoughts. Dr. Whiteman, I know you're still very much active after 50 years, and it's a real inspiration, the work that you continue to do. We really thank you for all your contributions and your perseverance and commitment to rare disease, both clinical care and drug development. It's a tremendous legacy. And, Jerry, I'm so glad you were able to share today. Yes, let's take a moment and celebrate that. Thank you for coming, sharing your thoughts as well. We will definitely all be keeping an eye on this space, and I'm sure in the future we'll have the opportunity to get back together and talk about mitochondrial disease therapeutic development in greater detail. But for today, I'm going to bring our Powerhouse podcast to a close for this episode. To the audience, thank you for watching. Please consider interacting with UMDF if you're a part of the patient family community. We're here to support you in any way that we can. And we hope to see you on a future episode of the Powerhouse podcast. Thanks, everybody.
Thank you.
Bye-bye.